Hello and welcome to the Nelson QCE Biology webinar. My name's Catherine, and alongside one of our authors, Wendy Cook, today we want to talk you through the practical strategies for implementing the new syllabus.
This is a sneak peek of all of the QCE science titles that will be available from Nelson. In today's session, we want to briefly cover the updates of the new syllabus of V1.1. Then Wendy will take you through the student experiment, how to think about it, how to plan it. And then I'd love to be able to show you how the Nelson QCE Biology Units 1 and 2 resource is able to support you and your students through the new syllabus.
Hi, my name is Wendy. I've been teaching, as the slide says, for over 27 years. I've taught in the UK and I've taught in Queensland. I've spent a lot of time with senior biology subjects, and I've been teaching this new syllabus since it was introduced in 2019. The next few slides give a summary of the changes to the syllabus, comparing the 2019 syllabus to this new syllabus that we start teaching from next year. I think the biggest change for me that I've noticed is the clades information going from Unit 3 solely to Unit 4 and the impact that has on the type of questions that we can put in a data test. Otherwise, the total level of content seems pretty similar to what it was previously, and therefore delivering the new syllabus presents the same challenges as the previous syllabus.
As you can see here, we have a summary of some of the updates that have been made from the new syllabus. Now, just to note, please, this is not a comprehensive, exhaustive list. What we've done is just highlighted some of the key changes that we've identified as part of the new syllabus. As you can see here for Unit 1, a few of the changes have been the movement of some of the dot points around topics and also the updates of some verbs used to describe some of those syllabus dot points, as well as the addition of some new content around the she's, around the inquiries, but also things around biomolecules, catabolism, and anabolism as well. For Unit 2, a little bit more content that's been removed slightly here. For example, osmoregulators, osmoconformers, as well as some virulence factors, as well as some immunity-related content around virulence factors, for example. Again, updates on some of the verbs used for the dot points in the syllabus, as well as some new she's and some new inquiries. In terms of Unit 3, some removal of content around species diversity, also cladograms, actually now moved to Unit 4, as well as some simple diagrams as well. As well as some simple flow diagrams. Now again, updates on some of the verbs that are used to describe some of the key content in the science understanding, and new she's, new inquiries, but also content around sampling techniques, as well as species interactions. In Unit 4, some of the content has been removed from the genetics topic around gene expression, on disjunction, as well as removal of content around some phylogenetic relationships. As we've seen for the other units and topics, updates on the verbs used for some of the science understanding syllabus dot points. And in terms of the additions, we can see more information around frameshift mutations, around interpreting profiles, DNA profiles, as well as I mentioned before in Unit 3, the movement of cladograms here into Unit 4, some new she's, and some new inquiries in general.
We've really tried to take an opportunity here to provide both teachers and students with suggestions for how a student experiment could be developed from the practicals listed within the textbook. As we all know, one of the key features that we're looking for with a student experiment is guiding students to meet the expectations of what's stated on the ISMG. There have been some changes to the ISMG, but overall the attributes that we're looking for students to present in a student experiment haven't really changed. So the new criteria names are forming, which includes a rationale, research question, and a methodology. Finding, which is the actual data collection. Analysing, interpreting, and evaluating, which are pretty similar to what we used previously. In the textbook, there are examples of research questions that are considered both specific and relevant. Not all of the practicals published have that opportunity. For example, the microscope section, some of those practicals are really about using skills and calculating magnification. So there's no investigation involved in those particular tasks. However, there are practicals that do give us the opportunity to put in examples of research questions that really focus on what the independent variable is, what the dependent variable is, how these are being measured, and what the context is for that research. So in the plant physiology chapter, there's a practical that looks at the presence or absence of light and what that does for photosynthesis. So this allows students to see how an independent variable can be written, how a dependent variable can be written, the units that the variables are being collected in. I'm going to do that again. So it shows how the independent variable, and again, it shows the independent variable and what is being measured, and often the units that those are being measured in. It then looks for stating what the dependent variable is and how that is being measured. And in this instance, you can make it even more specific by stating a type of plant being used. So the text example is provided here. And then below that is a research question that has been confirmed as both specific and relevant. And in this example, we can see that light intensity, the units are provided, the range of light intensities that are being covered are provided, that the dependent variable is gas bubble production and what unit that's being recorded in, and the specific plant species is being named. So students can actually take the research question that's provided either in the textbook or that they've worked on themselves and have some idea of how they would take that from being relevant, because most kids can generally get a relevant research question. It's making it specific that they find challenging. So we have hopefully provided some examples of those relevant research questions and those that are specific and relevant in the textbook. Sticking with forming, the justified modifications to the methodology. You'll notice that in a lot of the practicals, there's a prompt there for students to write down what variables have been controlled. And when students are asked to provide ways to modify the methodology and then justify those ways to develop a new research question, having an idea of the variables that they controlled in the original experiment can give them some prompts to think about ways that they can modify the investigation in the future, and then it can be linked to the new methodology in the form of a justification. So remembering that the modifications include refinement, redirection, and extension. So if kids understand what variables they controlled initially, it does give them an idea of ways that they can extend their investigation or actually refine their investigation if they weren't able to control those variables the first time round. Finding has three aspects. One is the use of scientific language. One is the actual collection of the data, making sure that you have sufficient and relevant data. And then the third aspect is the risk assessment. The practicals included in the textbook have these risks identified and addressed in the form of a table. This just gives students one way of being able to present this information. And where appropriate, the practicals have identified general risks that we typically have identified around chemicals or use of equipment. We also consider ethical issues and environmental issues as appropriate, and students can see ways of identifying and addressing these. The next criterion is analysing, where students are expected to be able to identify relevant trends, patterns, relationships, uncertainties, or limitations. You'll notice that the practicals that are included in the textbook include opportunities to collect both qualitative and quantitative data. So students have practice at collecting and organising both types of those data. In most cases, the instructions under the analysis heading direct students to presenting the data in some type of graph. Once a student has got the information into a graph, it's often easier for them to identify any trends or patterns or relationships that are present in the data. Chapter O, which is the online chapter, has examples of ways that students can process data to identify uncertainty, such as uncertainty of the mean or standard deviation. And it also has examples of ways that the data can be presented in different forms other than just straightforward graphs, more so probably for Unit 3 when you're looking at things like kite diagrams. Other skills that the kids are asked to do during the textbook practicals are averaging, which is something that everyone should be able to do, and then moving on to those measures of reliability. Beyond that, looking at R and R squared values and correlations and regressions and all those kinds of things, they're really up to what you expect your students to do that is relevant. The most important thing is that it has to be relevant to the type of data that has been collected. And this is an example of a student from my own school whose work was confirmed at 20 out of 20 for this particular task this year, and how they have chosen to present their data and use the error bars function through Excel, which they set to represent the uncertainty of the mean. And then they've used some of the statistical functions on Excel to generate other analysis statistics that can be discussed in an evaluation. I've split the final criterion into two slides, talking about interpreting first. The sections labelled interpretation in the textbook practicals provide prompt questions to allow students to provide explanations for what they found out. Often I find that my own students will give me just a really detailed description of the trends or patterns or relationships that they've already identified. So I think as many opportunities as we can have to get kids actually taking the scientific concepts that we want them to use to explain what they found out is really good. And so hopefully these prompt questions will give students an idea of what they need to include in an interpretation when they're producing their own report. Similarly, with the evaluation, under the evaluation heading, there are also prompt questions to help students to understand different ways that they can evaluate their data. And obviously, all ways to evaluate data are not present in every single practical, but across the range of practicals that are included in the textbook, students have the opportunity to consider lots of different factors about how to evaluate their data and the methodology that they've used to collect that data to make a meaningful evaluation of their work.
Now let's talk about the new Nelson QCE Units 1 and 2 Biology resource. How it can help support you as a teacher as well as your students to build the confidence throughout this new syllabus. Here's a look at the beautiful new covers for both the Year 11 Units 1 and 2 and Year 12 Units 3 and 4 book. We've made a lot of effort in this new edition to make sure that the content that we have in the book is clearly aligned to the syllabus. And as you can see at the start of every chapter, we actually outline all of the syllabus dot points that are associated and the content associated in the chapter. With regard to the theory content within the chapter, if there are any instances where we're going a bit above and beyond the syllabus dot point, we've actually noted that in the content as well. So we want to provide the opportunity for extension for students, knowing that it will deepen their understanding. However, also wanting to make teachers and students aware of the content that's a bit above what's required in the syllabus. We know that biology is all about making connections between different theories, different topics, across topics as well. And this is one of the key themes and the key struggles of students when it comes to the exams. And so we've made efforts throughout the book, both in questions as well as in theory content, to show the connections between different syllabus dot points. For example, in the questions, we have cross-chapter questions where students are answering questions that involve the theory content of the current chapter as well as a previous chapter to see the connections. Seeing the syllabus links within the theory content, as well as at the start of every chapter, there is a list of assumed knowledge, which is the information that students have learnt previously that will be relevant for their success in the current topic they're about to learn. As we all know, students need a lot of practice applying their understanding, but also in the context of what it might look like in an exam. And so we have exam-style questions at the end of every chapter, both multiple choice and short response, to help students develop that confidence and have plenty of practice. And where applicable, we've also modified previous exam questions so that students can further their practice. Biology is not just about theory, it's also about the implementation of skills. So we also have in every chapter data analysis questions to help students build the capacity to analyze data effectively, as well as consolidating the key science skills through the use of the practicals. For you as a teacher, we've built a whole host of different types of resources to help you plan, implement, and also assess the content, whether that be risk assessments, whether that be teacher planners, unit exams, and a bank of questions that you can use whenever you like to develop your own formative tests to see how the students are going. So if you want to know more about the Units 1 and 2 Biology resource or any of the other science resources that we have available, please feel free to reach out to both Amanda or David, your Queensland consultants. Thank you so much for your time today.